Good morning, everyone. We are going to wait a moment so that everybody can join us. And thank you very much for your patience. We won't be long. So once again, hello everyone, and welcome to the webinar on alternative techniques in microbiological analysis organized by Condalab. I am Aletia Cruz, product manager for microbiology, and I will carry out the presentation today. First of all, I would like to thank you for your interest in this webinar, as well as your assistance. It is a somewhat long presentation, so to try to avoid interruptions and have a seminar as smooth as possible, please send us your questions on the question box on the platform, and we will answer these at the end of the presentation. If we take long to answer these or we have to end the seminar, don't worry because all questions will be recorded. So if your question in particular wasn't answered, don't worry. We will answer these via email. So let's begin. Today at Condalab, we are presenting our Condachrome product line. To do so, we will follow this next table of contents. We will talk first about the alternative methods and the market trends, as well as the solutions we can offer you from Condalab. We will continue with a brief contextualization of the different applications and sectors in which our products can be used. To finish, we will review in detail about the products we will consider to be most important. We have an extensive catalog of products, so if you are interested in any that we do not describe during the presentation, please do not hesitate to request more information or visit the specific section of Condachrome on our website at www.condalab.com. We are all aware of the time and work it takes a microbiological analysis, either by conventional or even traditional methods. Due to this, more and more laboratories are joining in the implementation of alternative methods. Different market studies indicate a growth of 7.8% for rapid methods and compared to a 4% for traditional methods. And why is this, or what may be the reason? Speed mainly, since the need for fast and reliable results is increasing. Whether due to the need for a quick and effective clinical diagnosis that can save a patient's life, or the urgent release of short-lived products that allow the industry not to lose the few and very valuable days of commercialization that these types of food products have. Likewise, sensitivity, that is the detection capacity at low concentrations, and specificity, very important to avoid false positives, are important factors when implementing this type of method. The rapid methods that we know and that are currently being used range from molecular diagnosis by qPCR to detect pathogens to immunological tests, mass spectrometry such as MALDI-TOF, or even new techniques such as NGS, which is next generation sequencing, increasingly implanted in clinical diagnosis, and that it is beginning to appear even in food safety. For that, we will probably witness its implementation in the years to come. But let's remember that these techniques mentioned do not offer only advantages. First, not all laboratories are equipped and prepared to implement this type of technique, especially the later ones. A high initial investment is required, both to condition the laboratory and to acquire the different equipment and reagents, which is only a small part of it, because we must remember and not neglect the importance of training the technical team and that they must specialize in each technique. It is for these reasons that with Kondachrome, we want to offer you an intermediate solution with the advantages of alternative techniques, but adapted to any type of laboratory. No investment of specific and high-end equipment is required, and the same personnel who works daily in a microbiology laboratory will be fully prepared to use them. That being said, what are these benefits that Kondachrome brings as a highly effective alternative method? In just 24 hours, once the plate is inoculated, you can actually see your results. These growths, due to the Kondachrome technology, will be of specific colors and characteristics of each bacterial group that allow an identification with the naked eye. We know that experience is a degree, and even more, well, more like a PhD, and even more when we talk about microbiology. It is not easy to identify the characteristic colonies of a certain microorganism, but with Kondachrome, it will be easy even for the most inexperienced eyes. On the other hand, in certain culture media, you will be able to observe different bacteria of interest in a single plate, thus saving time in preparation of media and also space in the number of plates. Furthermore, our Kondachrome culture media have a higher sensitivity and selectivity than traditional culture media. And now you may be wondering, but how can just simple color detection be so effective? Well, Kondachrome is based on three fundamental pillars. Firstly, Kondalab culture media, secondly, chromogenic substrates, and finally, bacterial enzymatic reactions. 60 years as manufacturers of culture media have given Kondalab unparalleled experience in its manufacture and development, gaining extensive knowledge, meaning the know-how about the nutritional and selective components in all types of media. Once we have developed these selective culture media, we add the chromogenic substrates that are colorless compounds that when hydrolyzed release a chromophore into the medium. This chromophore is internalized by the bacteria, so the colony presents this unique and perfectly distinguishable color. And all of this could not happen without the specificity of enzymatic activities of the different bacterial groups. These enzymes hydrolyze the compound present in the medium in a specific way to release the chromophore that is internalized by the bacteria and gives the colony a unique color compared to the rest. If this has not yet convinced us fully of the suitability of the use of Kondachrome culture media, let's see the different examples of how this simple in quotation marks, mechanism gives us a specific, a higher specificity to our method. Beta-D-galactosidase is an enzyme characteristic of the coliform group the chromogenic substrate known as salmon gall is hydrolyzed by said enzyme, releasing the chromophore into the medium and giving these bacteria a pink or reddish color exclusive to this bacterial group. It should not be forgotten that the composition of the medium has been developed for the growth of these microorganisms, so it is already selective. Another strategy, which is used, for example, in the CCA medium, is the differential reaction against different chromogenic substrates. Escherichia coli being a coliform, it would hydrolyze salmon gall. How would we differentiate then from the rest? We add a second chromogenic substrate, X-gluc. This is hydrolyzed by beta-D glucuronidase, unique in E. coli and not the rest of coliforms. In this way, instead of giving a greenish blue to the presence of X-gluc or a pinkish color due to the presence of salmon gall, it will give rise to colonies of a very characteristic dark blue or violet color that will differentiate E. coli from the rest of coliforms. And this characteristic color comes from the hydrolysis of both substrates. Finally, we can also play with the inhibition of these enzymatic reactions. For example, in our Condachrome Vibrio, which we will review later, we would identify the growth of Vibrio alginolyticus from the rest of the species, because in this, the beta-galactosidase activity is inhibited by the high concentration of sugars in the culture medium, which is around 20% sucrose, giving rise to colorless colonies and differentiating them in this way from the rest of Vibrio species that grow in the medium with a greenish blue or a pale pink color. Once we have seen what alternative methods the market offers and what Condachrome can provide us, in this second section, we are going to see the different applications that it can have. We will understand as applications those sectors or types of analysis in which you can find a solution that suits you. We will do a little review of the current issues in them so that we can have an overview of the sector and see how Condachrome can help in these types of industries. So first, we have clinical diagnosis. That is a segment of the microbiology market with the greatest economic impact. Traditionally, by having more investments, especially in the public sector, than the rest of the industries, they have been incorporating new technologies and diagnostic methods before the rest even. Among these new technologies and techniques, the NGS stands out. These and increasing advanced massive sequencing systems at a competitive costs are revolutionizing the sector due to the number of applications that this technique offers, especially in pharmacogenetics or in drug development. So this technology and its implementation is becoming a common diagnostic tool. Another key point in the clinical sector is the appearance of diseases acquired during the hospital stay, that is, nosocomial diseases. Normally, on account of bacteria and viruses present in the hospital environment that take advantage of the immune weakness of the patient to infect them. It should not be forgotten that in these environments there is a great selective pressure due to the widespread use of antibiotics and antivirals in hospitals, which is evident. And this high selective pressure brings us to the next point. So if AIDS was the main public health concern in the 1980s, and today we can safely say it is cancer, resistance to antibiotics will be the great challenge that we will soon have to face. Every day there's more and more data on new resistance and less treatments to combat them. For this reason, the detection of these resistant species is essential and key to applying a good treatment strategy. With Chondrochrome, you can find culture media for the detection of bacterial resistance or microorganisms responsible for nosocomial diseases, such as Pseudomonas or Staphylococcus. Another very important sector where our Chondrochrome solutions can contribute to different laboratories is the food sector, that is, food safety. It is a fundamental sector for any country, which grows year after year. In addition, it has more and more conscious users who are looking for products with very different characteristics, but always with a common denominator, this being safety. Any small failure in delivering products, such as in poor conditions, wrongly labeled, or contaminated to the market, can make any company, big or small, lose all the reputation it has gained over the years, even forcing them to shut down in some serious cases. Within food security, the two main parameters analyzed in almost any food matrix are Listeria and Salmonella. And for this, we will find countless solutions in the market, including our line that we present today. But it is not necessary to analyze only these two microorganisms to guarantee food safety. Increasingly, there is an awareness of microorganisms that can pose a danger to public health and of which until now were not taken into account much. Campylobacter, for example, is the food poisoning with the highest worldwide prevalence, even above Salmonella. Another case, for example, can be the control of pathogenic strains of E. coli, such as STEC. Even Yersinia, a bacteria that has not been investigated extensively in the sector, occupies a position of higher incidence in foodborne infections, more than Listeria, according to the EFCA. Despite this data, it is true that the sector is highly regulated and there are still no stricter laws for these parameters that we have been reviewing. Although everything comes sooner rather than later, because in early 2018, a new hygiene parameter was established for Campylobacter in broiler carcasses that had not been previously contemplated. Lastly, the need of the industry in terms of quick results, given that, as we've mentioned before, many products have a short shelf life and traditional analysis can take anywhere from 5 to 15 days, depending on the parameter, immobilized material, and these loss of days on the market make companies prefer to have control approvals sooner. Nevertheless, in face of this need, reliability in the results must not be neglected or even given less importance, because what would be the use of rapid techniques that allow a batch to be released if later I have to withdraw the product from the market? It should be borne in mind that in many cases, confirmation by traditional means is necessary to consider the result as good. And finally, in which other sectors can Chondrochrome be useful to us? In the cosmetics sector, for example, we can carry out an analysis of the different pathogens in chromogenic media, obtaining thus clear results prior to confirmation. In water control, chromogenic media are beginning to be included in international standards such as ISO. And some of our products can simplify the protocol by using a single culture medium instead of two different media, such as the case of enterococci. Another important point is the control of biofilms, where, for example, like Listeria or Pseudomonas are major players in the formation of these bacterial microstructures, very common on work surface or even in water systems. These biofilms are directly related to the control of the production plant, given this can be many times the origin of possible contamination in the final product. Well, we have already talked first about the different alternative techniques on the market and how Condachrome can become a great ally. Also, we have made a review of the different applications in sectors and the challenges that from CondaLab we believe that these sectors face. Let's move on to the last part of the presentation. As we have said, our Condachrome catalog is quite extensive. Therefore, in this last part, we will go into detail of the 10 media that, in our opinion, have greater importance or relevance in the different applications mentioned above. So let's start. The first medium we present is Condachrome Utic, aimed at detecting agents that cause urinary tract infections. It is a medium exclusively for use in clinical diagnosis and has CE IVD marking. Now, in the same culture medium, we can identify, let's see if I can point at the presentation. Okay, but we can see here on the image, we can identify Enterobacter and Klebsiella that would grow in a dark blue. Enterococcus would be a lighter blue, and Escherichia coli would grow pink. Then we could have Salmonella and Pseudomonas in an amber color and Proteus in a light brown. This medium replaces the traditional method where 3 selective agar were used for identifying the infection: plate agar, McConkey, and also blood agar. This was a somewhat confusing method with a laborious process of preparation and with a complex reading, since the different species in these agars could not be identified, and it would be necessary to perform confirmation tests. For the procedure, it is very simple, starting with the urine sample streaked directly on the plate. Incubate at 37 degrees Celsius for 18 to 24 hours. Okay, next, culture medium is Condachrome candida, used for the analysis of Candida species, as you might guess by its name. These species are closely related to superficial oropharyngeal and urogenital infections. It is a medium aimed at clinical diagnosis. It has the CE IVD marking, although it can also be used in other industries where this pathogen is of interest, such as in the analysis of cosmetic products. In the same medium, we can identify different species of the Candida genus. Candida albicans will go green, which is at the bottom of the plate in this image. Candida tropicalis will form blue colonies, which is on the right of the plate. And Candida glabrata will grow at a light white with purple in the middle, which is on the left of the plate. On the other hand, if we happen to find pink or pale purple colonies, they will belong to the Candida krusei species. In this case, we would go from a savurodextrose agar with chloramphenicol plates together with the laborious identification test to a single culture medium. The same method, from a sample of clinical origin, we will streak in the Condachrome agar and we will incubate at 37 degrees Celsius, observing the growth at 24, 48, and 72 hours. Okay, on this occasion, we present a medium with great utility in the clinical routine, that is Condachrome MRSA. It is a culture medium for the identification of strains of Staphylococcus aureus resistant to methicillin. It has the CE IVD marking and it is intended for clinical diagnosis. As we have mentioned, the appearance of resistance is becoming increasingly common, and the normal procedure has multiple steps to identify it, being in these cases a quick treatment a key point in therapeutic success for controlling a possible pandemic. For example, for this particular case, methicillin resistance was usually performed with a salt and mannitol agar, along with the addition of oxacillin. Detection was made at least after 48 hours, and there were quite a few false positives and negatives due to the confusion caused by the interpretation of results. With our series of media for studying the most common resistances, you will not have these problems. A medium that is strict directly with the clinical sample and in which in 24 hours you can have the resistant bacteria identified without errors in the reading. We also have media for resistance to vancomycin, gram-negative carbapenemase bacteria, and gram-negative lactamase bacteria. Okay. Let's move on to another culture medium that we will find is Candachrome salmonella, a microorganism analyzed both in clinical diagnosis, it has the CE, IVD marking, and in food and water samples. Little will we discover new of this pathogen, as it is the second agent responsible for gastrointestinal infections in the world. There are a few times that we do not see notifications about product withdrawals due to salmonella contamination, despite being, along with listeria, the most wanted pathogen or search in food safety. In our Candachrome, different salmonellas will grow magenta. Other enterobacteria that may appear will grow colorless, as Proteus, for example, or greenish blue as Escherichia coli. Therefore, for the count of salmonella, we will take into account only the magenta colonies, as we can see here on the image. The procedure for clinical samples is quite simple, starting from fecal samples or digital rectal examination. Inoculate the prepared plate directly. Incubate at 37 degrees Celsius for 18 to 24 hours. Another possibility is to use Candachrome salmonella in food matrices and in water analysis. But keep in mind that the method is not accredited by an agency like AFNOR or IOAC. Even so, due to its predictive nature, it is interesting to use it as a secondary medium of isolation together with the XLD. The inclusion of secondary culture media is a choice of each laboratory according to the ISO 6579 method, which is for the detection of salmonella that begins with either pre-enrichment with buffered peptone water or a direct streaking in selective media, that is the MRSV agar, and in parallel with a second medium, the MKTTN. Subsequently, Candachrome salmonella and XLD would be used for the selective isolation. Here we have Candachrome E. coli 157, intended for the identification of enterohemorrhagic strains such as O157H7 of Escherichia coli. The serotype is mandatory to report and the main culprit for staph infections. The scope of application is both food and clinical, since it has the CE IVD marking. Colony reading is very simple due to the characteristic pale pink color of O157H7, as well as the high selectivity of the medium. We can find total inhibition of other groups of Enterobacteriaceae, Enterococci, and Staphylos. And if the application of the product is going to be for clinical diagnosis, the medium would be inoculated with fecal samples. incubating at 37 degrees Celsius for 18 to 24 hours. In the case of using it in food samples, it can be used in the same way as the Salmonella analysis. The same ISO 16654 method includes a second selective medium for parallel incubation together with the CT-SMAC. In clinical diagnosis, this medium replaces McConkey agar with sorbitol and cefixime, where confirmation of both the red and white colonies that appear on the plate is required. A much more complex process than the one we propose with Condachrome. So, well, we have another product, Condachrome Listeria. This culture medium is formulated according to the ISO 11290 method, so it can be incorporated into any accredited procedure on disease standard. It is a medium used in food safety where it is one of the most analyzed parameters together with Salmonella. In the culture medium, the Listeria species will grow a greenish blue that we can see here on the image. The X-glucosidose glucoside present in the medium is a substrate for the detection of the enzyme beta-glucosidase, common to all Listeria species, giving the colonies their blue color. Other organisms that possess this enzyme, for example, enterococci, are inhibited by the selective agents within the medium and also by the supplement. As we know, the results of the investigation of Listerias occur both in species and in monocytogenes. We will be able to differentiate both in the same medium thanks to the presence of the halo, which is formed by the precipitation of lecithin due to the action of lipase that is exclusive to Listeria monocytogenes. This medium is inclusive for serotypes 4B and 1,2A. For the detection method, after incubation in half freezer and subsequently in freezer, the Condachrome Listeria plate is streaked over the surface. Once inoculated, it is incubated at 37 degrees Celsius for 24 hours. If there is not enough growth, it can be left for another day on the stove without a problem. In the case of enumeration, it is streaked directly from the half freezer, and this must be done simultaneously together with the detection. We are almost done. Another medium included in our Condachrome line that is also in accordance to ISO is the CCA. In this case, it's the ISO 9308 method. This culture medium is indicated for analysis of drinking water. As we saw a couple slides ago, the coliform group presents the enzyme beta-D-galactosidase, which will lead to the growth of pink colonies by the hydrolysis of salmon gall. For its part, Escherichia coli will also degrade the X-GLUC, giving rise to deep dark blue or violet colonies, since it will degrade both compounds. Other bacterial species, such as Pseudomonas, can grow in the medium, and together with the positive beta-glucuronidase, they will grow colorless, as in the case of O157H7. The method describes consists of filtering the sample through membranes, which are placed on the CC agar plates and incubated at 36 degrees Celsius for approximately 24 hours. To avoid false positive results caused by oxidase positive bacteria, for example, in Aeromonas species, we must confirm bacterial colonies through an oxidase negative reaction. Therefore, the total coliform count would be made up of negative oxidase bacteria, positive beta-D galactosidase colonies, which are pink to red, and positive beta-D glucuronidase colonies, which are a dark blue to violet colonies that we can appreciate here on the image. Let's look at this product on the list, that is the Chondrochrome vibrio. The vibrio genus consists of microorganisms whose natural habitat is marine and river ecosystems. Vibrio species are primarily responsible for causing cholera and food poisoning in humans. The sector of application is food, water, and even clinical analysis. The colonies of Vibrio cholerae and Vibrio vulnificus grow pink, while Vibrio parahemolyticus will grow a teal color because it is a beta-D glucuronidase positive bacterium. The high presence of sucrose in the medium will inhibit the action of beta-D galactosidase in Vibrio aldinolyticus, forming thus large colorless colonies that we can see here on the left of the plate. As in other procedures, ISO 21872 method for the detection of Vibrio species in food and animal feed requires a second selective medium of the laboratory's choice. It is here where our Chondrochrome vibrio can be included, giving a preliminary analysis of the different species before confirmation. Parallel to the seeding in TCBS, this culture medium would be inoculated and incubated at 37 degrees Celsius for 24 hours. Here we have next the Chondrochrome MIA indicated for the identification of Enterococcus. The medium was developed as a one-step procedure that does not require a transfer of the membrane filter to another substrate. Using this method of single-step detection by membrane filtration, a wide range of sample volumes of fresh estuarine or marine drinking water, even drinking water, can be tested and enumerated. This culture medium is an improved formula since it allows the differentiation in the identification of the two most analyzed Enterococcus species that are Fecalis and Faecium. Enterococcus fecalis colonies will grow a greenish blue, and the Enterococcus faecium will have a very deep blue color, which are in the image the slightly larger colonies that we see. The procedure consists of filtering the sample through a membrane, which is then placed on the chromogenic agar, incubating at 41 degrees Celsius for 24 hours. In the case of other membrane filtration methods, a second selective medium would be required. Oops. So finally, as you had a preview, we have our final product that we present today, that is the Chondrochrome pseudomonas. Due to the ubiquity of these bacteria, which are present in a multitude of diverse environments, the applications are very wide. I range from analysis of meat products, analysis of water, cosmetics, to even clinical diagnosis. Pseudomonas is a genus that has a great impact also on the formation of biofilms, so its elimination and control is quite tricky. Furthermore, it is an opportunistic pathogen with great genetic plasticity. For these and other reasons, it is considered an emerging pathogen of great clinical importance, since it can infect almost any part of the human body, really, with the colonization of the respiratory tract being the most affected. In fact, Pseudomonas is the cause of almost 50% of nosocomial bacterial pneumonias. And genetic plasticity, that is the exchange of genetic material between individuals is a weapon to keep in mind when we talk about generating mechanisms of resistance to antibiotics, which is critical in environments with wide selective pressure, such as hospitals. The reading of the plate is very simple, since the species belonging to the genus Pseudomonas will grow magenta and has great inhibition for the rest of microorganisms. In the case of any growth appearing, it would be colorless. The method is again quite simple, starting from the sample or its dilution, depending on whether it is for clinical or for other sectors. It is strict and incubated at 35 degrees Celsius for 24 hours. And if necessary, it can be left up to 48 hours to obtain more defined colonies. So with this, we finish the presentation. I hope you have found this webinar interesting and that we have encouraged you to implement chromogenic methods in the laboratory. The benefits are extensive for the little investment they require, and although I know that the little can be relative, always depending on what we compare it to. Chrome offers you quickness and simplicity in your methodology with a much lower investment than other rapid techniques on the market. I leave you now with our video that lasts very little, and then we will begin with the round of questions. So let's see.
Specific color that makes it possible to identify the bacteria at first sight. Advantages. After only 24 hours, you can collect the results in the culture medium. Identify by color at a glance the microorganism grown in the culture medium. Save time by identifying different bacterial species in the same culture medium. From the sample or from your specific enrichment broth. Variety for a multitude of microorganisms. At disposal, culture media for clinical diagnosis, antibiotic-resistant microorganisms, cosmetic products, food safety, water control, and production. Step into the next generation of culture media.
Okay, I hope you have enjoyed the video, and now we will go on to answer some questions. We are already running on time, so out of time, so we will try to do it really fast. Okay, let's see. Okay, so we have a question. Does samples in food industry need pre-enrichment for chromogenic testing? Usually it depends on the sample and almost exclusively on the situation. If you have a sample with a high content of the microorganism of interest or not. Usually, as I've mentioned, it can be strict directly. You don't have to dilute, but if it has a lot of concentration, it is more commonly diluted to have a better plate reading. So it depends fully on the samples, but it is not an obligated requirement. Okay, we have another one. What is the difference between the Kondachrome MRSA and the modified chromogenic MRSA? Okay, so with the Kondachrome MRSA, as we have mentioned, we can carry out the detection of methicillin-resistant strains of Staphylococcus aureus, and it is that a straightforward culture medium for identification of these strains. So then, by making an improvement, the modified Kondachrome MRSA is able to detect and differentiate between Staphylococcus aureus and Staphylococcus epidermis, both methicillin-resistant strains. And in this medium, Staphylococcus aureus will grow as magenta colonies, and Staphylococcus epidermis will grow as a greenish-blue colony. Also, the rest of the accompanying flora or microorganisms will be inhibited. Okay. Okay, many are asking, okay, it is a very common request, so I will answer it quickly, and then we will end the presentation. You are requesting if you can have the presentation slides, because it is very helpful. We love that you have found it very helpful. You should receive the video, the recorded video of this presentation, so you will have it via a video, not the slides as such, but you will have the video. Our marketing department will send you the video link, the link to the video, so you can watch it as many times as you'd like. Okay, so I don't want to extend any further. I know that it is early in the morning. We have a lot of things to do more. So the few questions that we have not answered, once again, we will answer them via email as soon as possible. So please don't worry. We want to, again, thank you for attending this webinar. And any other questions or comments, please contact us via your area manager or via our website. And as always, we hope to see you in the next webinar. Thank you.